Have you ever wondered how we can peek into the bustling, silent world inside our heads to hear the brain's very own symphony of thoughts and feelings? It is an intricate dance of electrical impulses. Electroencephalography, or EEG, measures these brain electrical signals by detecting the summed electrical activity of large groups of neurons through electrodes placed on the scalp. It essentially acts like a super-sensitive microphone, listening to the collective hum of millions of brain cells communicating. The fundamental principle behind EEG lies in the electrical impulses generated by neurons. Specifically, it primarily records postsynaptic potentials, which are voltage changes occurring on the dendrites and cell bodies of neurons. When many neurons, particularly those in the cerebral cortex, are active in a synchronized manner, the ionic currents flowing across their membranes create an electric field that extends beyond the individual cell. This electrical activity then propagates through brain tissue, cerebrospinal fluid, and the skull and scalp. While individual neuronal firings are too small to detect from the surface, the synchronous activity of tens of thousands to millions of neurons generates a sufficiently strong and coherent electrical field that can be picked up by external sensors. This phenomenon is known as volume conduction. EEG electrodes, which are small metal discs typically made of silver chloride, are placed on the scalp using a conductive gel to ensure good electrical contact. Each electrode picks up the minute voltage fluctuations at its specific location relative to a reference electrode. These tiny voltage differences, often in the microvolt range, are then amplified many thousands of times and recorded. The spatial arrangement of the electrodes, often following an international 1020 system, allows researchers to infer the approximate location of the underlying brain activity. The EEG signal itself is a complex waveform reflecting the oscillation of these neuronal populations at various frequencies. For example, specific frequency bands, such as alpha waves between 8 and 13 Hz, beta waves between 13 and 30 Hz, theta waves between 4 and 7 Hz, and delta waves below 4 Hz, are associated with different brain states and cognitive processes. Neuroscientists analyze these patterns, their amplitude, and their synchronicity to understand brain function and identify abnormalities. The raw EEG data is then processed using advanced signal processing techniques to extract meaningful information, such as event-related potentials, which are brain responses time-locked to a specific stimulus or event. Ultimately, EEG provides a non-invasive window into the brain's dynamic electrical landscape, offering invaluable insights into cognitive processes, sleep stages, and neurological conditions. By understanding how these electrical signals are generated and measured, we gain a deeper appreciation for the brain's complex mechanisms and unlock pathways for both fundamental research and clinical applications.